we're happy to have Dr. Manuel Rosa-Garrido here. Dr. Rosa-Garrido studied in Spain, biology and biochemistry, to then start the PhD at the University of Cantabria in biomedicine, studying how the epigenetic affects cancer progression. He then moved, after getting the PhD cum laude, he moved to the University of California in the US to study cancer epigenetics, and in particular how CTCF is involved in heart failure. And this work led to the was supported by a postdoctoral fellowship from the American Heart Association. He was awarded several prizes, and all this work led to him being appointed as assistant project scientist at the David Geffen School of Medicine, where he is now working on new epigenetic therapies for cardiac diseases. So we're happy to have you here. The floor is yours.
Okay, so thank you very much for the kind introduction, and yeah, thank you also for giving me the opportunity to share all our research with you guys in such a nice seminars. So today I'm going to talk to you about this project we have in the lab when we study temporal chromatin remodeling during cardiac disease. Okay? So cardiac disease is a pathology where the heart increases its size to try to compensate the lack of activity in an initial phase called hypertrophy. So hypertrophy is then a natural response of the heart to try to balance the entire system. So hypertrophy is characterized by the increase of size of the cardiac cells and increase of the heart wall thickness. So hypertrophy can be reversible in some cases, like pregnancy or the cases of the professional athletes when they stop the training. The problem is that if we keep hypertrophy for a very long time, the heart is not going to be able to keep this condition, and it's going to enter in heart failure, okay? Where the cardiac cells start dying and the heart wall thickness get reduced. So during this process, development of hypertrophy and heart failure, there is a change in gene expression profile that is controlled by different epigenetic factors like DNA methylation, chromatinaccessibility, histone marks, and the activity of different transcription factors, activator and repressors. So these factors act locally and constitute what we call chromatin remodeling. So we got interested in studying chromatin remodeling during the set of heart failure because in our previous work, we saw that even in the epigenetic environments organized by high-order chromatin structure, you can find transcriptional exceptions. So you can find inactive genes in active environments and active genes in inactive environments. So to try to understand if changes at the local scale, like chromatin remodeling, can explain these exceptions, we decided to start studying DNA methylation and chromatinaccessibility by performing RRBS, reduced representation basiophil sequencing, and ATAC-seq. So when we started this project, then we decided to use two different models of heart failure. The first one that we tried, the first one that we used was transverse aortic constriction, TAC. So TAC is very well-known pressure overload model where performing a small knot in the aorta of the animal, a mouse in this case, you are going to be able to induce hypertrophy or heart failure. So to also study the temporal nature of the epigenomic compensation in this model, we perform all this experiment at 2 time points: 3 days after TAC, that is the early compensation phase of cardiac pathology, and 3 weeks after TAC, that represent the decompensated phase of the disease. So the second model that we use, it was a specific cardiac-specific knockout for the protein CTCF. Okay, so the CTCF is a multifunctional chromatin regulator able to bind along the entire genome by using different combination of its 11 zinc fingers. So the main activity of this protein is to mediate genome organization, especially by mediating chromatin looping. That is this process by which regions that are far from each other in the same or in different chromosome get close together. Okay, so as I mentioned before, in our previous experiment, in our previous project, what we found is that when you remove CTCF from the heart of the animals, these mice develop a very strong disease phenotype characterized by dilation of the left ventricle in both diastole and systole, and dramatic reduction of ejection fraction. That is the amount of blood that is pumped out of the heart. Okay? So the heart of this animal is very big and is not working very well. So we use it as a model of heart failure. So what we did then in this project was to induce heart failure by depleting CTCF or performing TAC, wait until the heart was sick, then isolate the left ventricle, and use it to perform bisulfite sequencing to study DNA methylation, and perform a TAC-seq to study chromatin accessibility. So when we got the data, the first thing that we found is that what we found is that there was a widespread change in chromatin accessibility after 3 days that stabilizes after 3 weeks. If you compare 3 days and 3 weeks, you can see that the changes in chromatin accessibility detected by our experiments are moving toward the chromatin accessibility, suggesting that the genome gets more open as the cardiac disease progresses. So when you study the chromatin accessibility in the knockout animal, in the CTCF knockout animal, we also find big changes in chromatin accessibility, but in this case, the changes move toward the repression, the close of the genome, okay? This one makes sense because it's well known that CTCF act as a barrier of the heterochromatin. So if you remove CTCF, you're going to have more heterochromatin. So when we study DNA methylation, what we found was that there were more differentially methylated CpGs at late phase disease, after 3 weeks TAC or CTCF knockout, than at early phase disease, 3 days, okay? But in the 3 cases, the tendency of the genome is to get hypermethylated, to lose methylation. Next thing that we did was to compare the SER ataxic peaks and SER CpGs detected by our experiments. So when we did this thing, we found that only 28% of the ataxic peak with detected change in chromatin accessibility were SER between the early and the late phase disease, okay, 3 days and 3 weeks, suggesting that there is a time-dependent shift in chromatin accessibility during development of cardiac disease. When we do the same, comparing the 2 samples at late phase disease, 3 weeks and CTCF knockout, we found that there were 50% of these ataxic peaks were shared between these 2 samples, suggesting then that the cardiac disease presents similarities at late phase of the pathology, independently of the stimuli that we use to induce the disease. When we study DNA methylation, what we found is that 65% of differentially methylated CpGs were shared between early and late phase disease, suggesting that changes in DNA methylation are established at early phase and are well preserved during the progression of the cardiac pathology. When we compare the 2 samples at the late phase disease, we found 56% of similarity between the differentially expressed CpGs, suggesting again that cardiac disease presents similarities at late phase of the pathology, independently of the stimuli that we use to induce the disease. Next thing that we did then was to correlate these 2 features, chromatin accessibility and DNA methylation, with changes in gene expression. So when we perform gene ontology on these genes that are differentially expressed with detected change in chromatin accessibility or DNA methylation at their promoters or gene bodies, we found very similar terms related to neurogenesis, heart development, or organization of the cytoskeleton. When we now study the correlation between chromatin accessibility and DNA methylation and gene expression, what we found is that while the CTCF knockout sample follow the expected pattern, upregulated genes are more accessible and they are hypomethylated, and downregulated genes are less accessible and they are hypermethylated, the TAC 3 weeks doesn't follow this pattern at all, okay? Suggesting that there is something else involved in the regulation of these genes. So interestingly, when we studied the location of regions with altered accessibility or DNA methylation, we found that the majority of these regions are located in intergenic or intronic regions. Intergenic or intronic regions, yes. So this data suggests that the modification of these 2 features in these regions can be affecting regulatory elements like enhancers that are the ones that are controlling the expression of the genes. So to test this hypothesis, the first thing that we did was to use already published list of cardiac enhancer and combine this list with our Hi-C data to figure out what gene each of these enhancers was interacting with. So the next thing that we did was to test changes, to check the changes in chromatin accessibility and DNA methylation at the level of the enhancers and correlate these changes with the expression of the genes that the enhancer is interacting with. Okay? So when we did this thing, the first thing we did was to check methylation. Okay, so when we check methylation at the enhancer, we found that even when the changes at methylation at enhancers are 100% in almost all the cases, meaning that the enhancer is always methylated or always demethylated, this feature is not able to predict the activity of the gene that the enhancer is interacting with. So you still have hypermethylated enhancer interacting with upregulated genes and hypomethylated enhancers interacting with downregulated genes. Okay? So to test if there are other epigenetic factors involved in this process, we perform this experiment where we study occupancy of different transcription factor and histone marks at both the enhancers and the promoter of the gene. Okay? So this experiment detected binding of different cardiac key regulators like GATA4, TBX5, TED, the coactivator B300, and different active histone marks, suggesting that the regulation of gene expression mediated by methylation of the enhancer frequently requires the coordination with other epigenetic factors, like this one. So when we study changes in chromatin accessibility, what we found is that the changes detected at the late phase disease were already committed at early phase disease. Okay? So this behavior was also found at the transcriptional level. So you can see here how changes at late phase disease, 3 weeks and CTCF knockout, changes in gene expression at late phase disease were already primed at 3 days, okay, at early phase disease. When we check change chromatin accessibility at the level of the promoters, we found a similar behavior. So here you can see again how changes in chromatin accessibility detected at late phase disease at the level of the promoter were already established at early phase disease, 3 days. When we now correlate these changes with gene expression, what we found is that in the 64.4% of the cases, the gene is following the expected pattern. So downregulated genes have less accessible promoter, upregulated genes have more accessible promoters. If you perform gene ontology in this gene list, you are going to have, we got terms related to organization of cytoskeleton, heart contraction, and heart contraction. So one example of a gene of this list is ATP2A2. It's very well known. It's a gene that is very well known that is downregulated during development of heart failure. So you can see here what we detected at the promoter of this gene is that the chromatin accessibility decreases. When we study then chromatin accessibility at the level of the enhancer, we found the same behavior. So here again, you can see how changes in chromatin accessibility detected at the late phase disease at the level of the enhancer were already committed at early phase disease. When we compare, when we correlate these changes with gene expression, we found again that in the majority of the cases, 65.5% of the cases, the gene is following the same pattern, the expected pattern. So downregulated genes interact with less accessible enhancer, upregulated genes interact with more accessible enhancers. The gene ontology of these genes generate terms, cardiac-related terms. Okay? To also study if there are other epigenetic factors involved in controlling the expression of these genes, we did exactly the same experiment that we did with methylation. So in this case, we found binding of GATA4, NKX 2.5, TED, and P300 at both the enhancer and the promoter of these genes, suggesting then that these epigenetic factors also participate in regulating the expression of the genes in these cases where changes in chromatin accessibility at the level of the enhancers are not able to predict the expression of the genes. So as a summary then, what I showed you is that early and late stage of cardiac disease are associated with widespread changes in chromatin accessibility and DNA methylation affecting gene expression. I also showed you that changes in DNA methylation are established early in disease and are preserved during late state pathology. I also showed you that chromatin accessibility and DNA methylation at the enhancer play a role in controlling gene expression during heart failure, but they are imperfect predictors of transcriptional behavior. And finally, we also saw that late stage changes in chromatin accessibility are established at early phase and amplified during pathology. Finally, I would like to mention that the findings that the results that we got in this project have 2 important implications. The first one is related to the early detection of cardiac pathology. So since we know that the change in chromatin remodeling rapidly respond to pathological changes, these changes may be used as a potential marker to predict cardiac disease. The second implication is related to the epigenetic treatment of cardiac disease. So since we know that these changes in chromatin structure are happening at the early phase of cardiac disease, the epigenetic reversion of these features may constitute a valid treatment to prevent the advance of pathology. So that's the two things that I'm working on right now. And finally, I would like to thank everybody in the Vondriska lab, especially Dr. Douglas Chapsky and Dr. Thomas Vondriska. And of course, I want to thank all our collaborators at UCLA, Dr. Yibing Wong, Vincent Ren, Matteo Pellegrini, and Marco Morsello. So that's all. Thank you.
Thank you, Manuel. Very, very nice talk. So I will go with the first question we have here. So I hope I'm pronouncing this correctly. So El Shahima is asking, have you explored histone acetylation mark readers that can also recognize methylated cytosines and its relation to the disease progression?
So yes, so we didn't do that directly, but yeah, there is a lot of people in the field that have work with histone deacetylase and histone deacetylase and histone acetylase, and what the people know is that during heart failure you have activation of histone deacetylases, So we didn't do that directly, but yeah, it's a thing that people have done in the field before.
Okay. There's another question that is asking, why do you think there are alteration in genes involved in neural development?
Yeah, that's an excellent question, and I don't know the answer. I'm super excited about this result. I don't know why. Now we are trying to figure out if it's something related with the neurons that you can find in the heart, but we don't have any result yet. But we are super excited with this experiment, yeah, because every time that we perform gene ontology, we have neurogenesis there, So I don't know exactly what is happening, but we are working on it.
Maybe partially related to that. Have you so you've looked at a lot of cardiac enhancers in your data set, of course. I was wondering whether you had any if you looked at any enhancers of different cell types. So especially if the CTCF knockout leads just to deregulation of cardiac specific genes, or you see the emergence of enhancer that mark different cell types.
Yeah, so because the CTCF knockout is cardiac specific, yeah, the majority of the genes that we've seen affected are cardiac genes, But in the list of the RNA-seq, yeah, we still have genes that involve in neurogenesis is a lot, and also in organization of cytoskeleton, That is another thing that that make me very excited, because it means that the morphology of the cardiomyocyte is also involved in all this process, of establishing heart failure. So that's another direction that we are taking now in the lab. So that's in the future. But
very, very cool. Maybe I can have just a quick last one. We have another one, so I'll keep mine for later. Do you think accessibility changes in enhancer and promoter have different kinetics?
Yes, correct. Yeah, yes. So what we have seen is that yes, enhancer change before than promoters do. Yeah. Based on our results, what we see is that methylation at enhancer changes before that the methylation at the promoters. Yeah.
Okay, perfect. So thanks a lot once again.